All right. Yeah, thank you, Kristen, and thanks, Kevin, for hooking me up with this. It's a very interesting disease that we'll discuss today, beech leaf disease, and happy to talk about it. If anybody's interested in listening. So I'm Andrew Loyd. I am a plant pathologist researcher for the Bartlett Tree Research Lab. We are the research lab for the Bartlett Tree Experts Company, and I work on a variety of diseases of landscape shrubs as well as trees. And today I'll focus only on beech leaf disease and really my observations as well as some recent publications that have come out from people that are working on the nuts and bolts of the science of this, and hopefully give you a broad understanding of what's going on and what we're doing to try to find management solutions. So as probably a lot of you are familiar, let's see if I can, it won't let me, oh, there we go. This disease started to become observed in the Cleveland area in Lake County, Ohio in about 2012. So it's a fairly new problem. People started noticing the symptoms, which I'll go over in a bit, in that general area. And up until 2018, really had no idea what was going on or what was causing this disease. So what people were seeing were this very distinctive symptom. And the symptom is these dark banding patterns. Sometimes they can be chlorotic. Later in the season, they can sometimes turn brown. And it's really easy to see from the underside of the beech leaf looking up towards the sky. And so there's nothing else on beech trees that really looks like this. It's an easy one to diagnose in the field. But this is what you're viewing. And seeing it on other, here's another example in the forest where you have this banding pattern being shown. There it is again. It's pretty distinct. It can also occur on European beech in landscapes. So this disease is in natural areas primarily, and it's also coming on landscapes. But in landscapes, the European beech is probably more planted species, and it affects all cultivars of that as well. And here it is on a fernleaf beech, and you can see the banding pattern is not exactly the same, but it does still fit kind of that same similar symptom. Here it is on a European beech in Canada, and it shows that more yellow distinctive banding pattern. Again, it can be dark green to yellow or brown between the leaves, and this is on a hedge cultivar. And then as the disease seemed to progress after several years of successive infection, the leaves would start to come out and they would look crinkled or distorted like you see in this picture. Almost like similar to what we see with herbicide damage a lot of times. So this is something you see later in the disease after it's been infested for quite some time. And then here it is on a European beech. You can see them very malformed. And the galls are those banding patterns that are slightly raised, so we consider them gall-like, are present there in addition to the contortion or the malformed growth. Here it is again, showing that same symptom. And as going further into years of being infected, the tree starts to become sparse in the canopy. So they don't produce as many leaves. The leaves just kind of look sickly. And here's an example of that, the sparse canopy, not many leaves. And here's an example of a tree that has a lot of dieback. And not a lot of leaf production, and it's kind of this decline that occurs once you start to see the symptoms, the original banding pattern, to the crinkled leaves, to eventually this. And at this point, you know, you're dealing with other stressors as well, bark beetles, ambrosia beetles, canker, secondary canker pathogens like hypoxylin and things like that. And eventually the tree gives up and dies. So that, you know, observed trees dying in the forest as early as 6 years since initial symptom onset. And the smaller the tree is, the faster it dies. So this is something that is going to become more and more concerning, especially in the areas that have been hit hard. And we'll talk about the distribution here in a second. So this disease got a lot of press. You know, there were a lot of common press articles that came out, like this one from January 2019, talking about this mysterious disease killing beech trees. And it was, and it rapidly spreading, rapidly killing smaller trees, as I mentioned. And as you're probably aware, beech do propagate via saplings, and so that's how the regeneration most of the time occurs. And those small trees aren't even given a chance because they get infected and then they eventually die. So the larger trees are dying slower, but they're also being injured as well. But we don't know what, this whole mysterious aspect of it is something that has been talked about a lot. And so people don't really know what was causing it or whatever. So what do we know about it now in 2020 of September? So what we do know is that in Japan, there was a nematode that was described for the first time on Fagus crenata, Japanese beech, and they called it a leaf gall nematode, and they'd assigned the species Littlelinchus crenata to this nematode. And the symptoms of this in Japan on Fagus crenata are very, very similar to what was being seen in Lake County, Ohio, and other places. So you see this banding pattern on the leaves. It's discolored between the veins. It's somewhat raised and gall-like. And they describe these nematodes that were causing this symptom. And around the same time of this publication, a pathologist in Ohio found nematodes in the samples from Ohio, and a lot of them. And so they contacted the right people at the USDA. Dr. Lynn Carter from the USDA ARS, she's a nematologist, and they started to work on what is this nematode that's associated with these symptoms in the US and Canada. And so what came out of it in 2019 was they described a subspecies of the nematode from Japan, so Littlelinchus crenata mccannii. And this paper came out and described the species as well as the disease in the areas that were affected. So mccannii, the subspecies, is named after the pathologist David McCann, who identified these nematodes to begin with. So this is what the adult male nematode looks like that causes beech leaf disease or is associated with those symptoms. And it was described in 2019. So there's also this whole relationship of, does it cause the disease or is it associated with the symptom, was kind of the question. So one of the things that you do in pathology is you conduct Koch's postulates, where you take the associated suspect pathogen and then you put it into a plant. And so David Burke's lab, Dr. David Burke at the Holden Arboretum, worked with Lynn Carter and others, and they inoculated beech trees as well as with the Forest Service. And they inoculated buds by wounding them with a small needle and then putting nematodes, eggs, and adults into the bud with a syringe, parafilmed it or wrapped it up, or they infected leaves that had come out. They wounded those, put the nematodes in, and then monitored for symptoms over time. And what they found was when they inoculated these trees that were healthy otherwise, the leaves came out, if they inoculated the buds, with the symptoms that they were seeing in the forest. So this confirms that this nematode not only is associated with these symptoms, but is in fact causing them. And there is some talk about other, maybe it vectors another type of organism. That may be true. We haven't found it yet, but there's more data that hasn't been published yet that is suggesting that the nematode itself is causing this damage, and it is the primary causal agent of this disease. What they didn't find was when they infected leaves that they had wounded, they did not find symptoms develop. So this suggests that the buds, and the buds alone, are the infection court, or the area where the infections occur from year to year. So somehow these nematodes get in the buds, they cause the damage while in the bud, and then the symptoms come out in the spring. And that's another thing that Cleveland Metro Parks did a lot of observational work, and they followed individual leaves over time after they broke bud that had the disease to see if the symptoms progressed, meaning this banding pattern comes out like this, does it move into the adjacent part of the leaf, or does it stay there for the entirety of the season? And what they found is it does not progress. All of the symptoms that come out in the spring are the symptoms that are there by the end of the summer. So that's telling us a lot of information to provide that the buds is where the infections are occurring, where the damage is occurring while the leaf is still in the bud. And here's a picture from that paper from Lynn Carta and others, showing a bud that has been dissected apart with the bud sheets out, and you can see where the rectangle is there's a nematode in there. And I've extracted from some of these buds, and they're right underneath the bud sheets. You'll find in the wintertime, hundreds, if not thousands of nematodes in those buds, just waiting to cause the problem on the immature forming leaves. And it starts to make sense. We go back to these original symptoms that we're talking about, that the damage is occurring in the bud, because you can see that it's almost a mirror pattern of where the symptoms are occurring on the leaf. And that leaf was rolled up tight, and those nematodes were associated with the areas that when they were rolled up. So the symptom starts to make sense that the infections are occurring in the bud as well. And as far as the life cycle of the nematode and when the nematodes are active, so we know that nematodes are in buds, but then they kind of disappear. You can't find swimming in microscopic worms in the leaves when they first come out. They're hard to find, and the dates are not shown on this graph here, but this is May or early spring all the way through about October. And this is the number of nematodes that were collected in a given sample. So the average number of nematodes collected. So you can see as the season progresses, the growing season, the majority, the most nematodes are found in about August, September timeframe. So that's when they're active, they're swimming, and we think probably also moving in the plant. So microscopic worms, nematodes, they require films of water to move around. They can't move around on their own. They don't, fly or anything. So they would need something to vector them, or they would have to have a film of water to swim up the plant to neighboring buds to infect. And this pattern, again, May through October, starts to make sense, because when do the buds start forming? They form very early as the leaves come out, and they start to mature, and then they overwinter. And when they're mature, this is when these nematodes are most active. This is probably when they're going in, and they overwinter in the bud, and probably have sexual reproduction, and then lay the eggs into the leaves when they come out, and continue the life cycle the following season. So this is what the nematode does. So what, this nematode is causing this symptom, but what is it actually doing? So this is a microscope image. The top part of the healthy leaf up top is the cuticle, and then you have all of the mesophyll cells in the middle, and then you have the underside of the leaf. So this is looking at a leaf from a cross-sectional viewpoint. And so this is what a healthy leaf should look like in a beech sample. Well, when it's infected with these nematodes, you can see that these nematodes are actually consuming and eating all of these cells in here. So when that happens, photosynthesis goes way down, and you lose a lot of sugars that could have been made by the plant to allow it to overwinter well and produce a nice flush of leaves the following season. So that's what the nematode does. Now, the strategy of it, does the nematode produce some sort of toxin? Does it have some sort of microbe that helps it digest? There seems to be some evidence that there may be some gut-associated bacteria that help it digest some of the cellulose, but we think the nematode is doing this mostly on its own. So the disease overall, how does it kill the tree? What we think happens now, and we're constantly learning about this, is that you reduce photosynthesis in one season, and over years of infection, you're reducing it even more, and so it starves the plant out, if we want to make an analogy. So over time, the plant isn't producing as many sugars, it can't send them down to the roots, and then those buds get less every year that they are infected. And if you know this, beech trees have mycorrhizal associations, in particular ectomycorrhizae forming fungi associations, and it's possible that these partnerships are also skewed. We don't know this. This is something that we want to look at, but it makes sense that if the plant's providing this symbiotic fungus with sugars to help it mine for phosphorus and other elements in the soil and water, it's also probably losing a little bit because the plant's not producing as much. So it's likely that these beech are not going to survive as well if the symbionts, the mutualistic symbionts, are not working as hard either. So that's kind of how this disease is working, we think. So we started in Lake County, Ohio, and in Ontario, and this is a map of the distribution as of 2020 when this publication came out. So you can see it's spread through Pennsylvania. It's moving southerly in Pennsylvania. It's now in Connecticut and New York on Long Island, New York City, Stamford, Connecticut. And we know now that it's also, it hasn't been published yet, but it's in Massachusetts and Rhode Island. So this thing continues to spread. And we're really not sure exactly how it spread, how it was originally introduced. And we do think that it is an introduced nematode from Asia, sharing so much genetic and morphological similarity to the nematode described on Japanese beech, although some slightly different. But we think it's introduced. But how is it spreading this far? We do have some documented cases where we think it's been spread in the nursery trade, but the nursery people are aware of this thing now, and they have some state and local ordinances to not move plants out of infected areas. There is no quarantine to date federally, but the states are working to try to keep it local as best they can. But it's still spreading naturally, and we haven't figured that out exactly yet. So what we don't know is we don't know what vectors it. As I mentioned, nematodes, require water to move locally, but they don't have wings, they don't fly, so they require something is moving them around, especially since it's moving around so well in natural areas. It's not necessarily moving in landscapes. If it was only in landscapes, we would think, yes, definitely the nursery, but it's moving in these natural areas. So something in the natural areas is moving it around. And so this was a poster from a plant pathology meeting this summer from Dr. Lynn Carta, and they've started working with other folks, people that study mites as well as people that study birds, because they received several samples where they found these mites, and on this mite that looks green, it's dyed, it's entangled with all these nematodes, Litholinchus crenata. And so they had this happen on several examples where these mites seem to be carrying the nematodes. So that makes sense somewhat locally. These mites don't have wings either, so they're moving around by crawling, and maybe they can spread the nematode locally. But how does it spread on larger geographic distances, or even within a forest scale? And they started to look at these mites, and they started to see that some of these mites are maybe associated with birds. Birds have a lot of mites that are associated with them. They live in their feathers and in their nests. And there's a group of mites, in particular, or birds, excuse me, in particular the purple finch, that actually feed on beech buds. So again, remembering that the infection cord, or where the infection happens, it is in the bud. And these purple finches actually, that's what they feed on in the winter time when those buds are nice and plump. So it's possible that this is potentially a means of transmission, whether it's the bird itself, maybe it feeds on buds that have the nematodes, and its excrement moves onto beech trees that were naive or healthy, or in fact, maybe there are mites that also have the nematode, they're associated with these birds, and they hitch a ride. So we're still learning about this. There is a group of people that are studying the bird relationship. But what really got me interested and excited about this piece of work was I talked to Dr. Lynn Carta, and she told me to Google the purple finch range, or where it's distributed, because it does migrate a little bit. And this is a map that I found on Google, where you look at the permanent resident status, meaning it's there year-round, breeding resident status, and then possible migration in non-breeding areas. So where we know this disease occurs is in the permanent resident status of purple finches, as well as the breeding resident status. So again, if we think about this bird being prevalent in all of the areas where we found the disease so far, it kind of starts to make sense. I mean, maybe we're making it make sense by investigating this, but I really think it requires further work and looking at these birds a little better. Well, and that's what we know up to this point, and that's the one hypothesis that seems to hold up and make some sort of sense, regardless of having the data to support it yet. So again, here's the range of where we know beech leaf disease is, and if we look at this map again, we have northeastern Ohio, where it started, all the way over to New York, Connecticut, Massachusetts, and then into Canada, and that distribution of the purple finch lines up quite well. So what we also don't know about is, we're seeing this thing, it's being reported in new places constantly. How do we manage it? This is an area that often gets left behind as we're studying it. We're putting, potential regulations in place. We're trying to figure out the disease cycle. That's all important stuff, and everything that we've talked about is important. But as far as management goes, how do we manage this, whether it's preventively or therapeutically? And so we started to look at that. And we wanted to look at products, that were already labeled, specifically that might have nematocidal properties and labeled for trees. And so just to give you an example of two chemistries here, the one on the right is associated with heartguard. So heartworms that infect our pets are nematodes, microscopic worms, and veterinarians prescribe this ivermectin chemical. So this is what the chemical structure looks like. Might look a little scary to look at these things. You haven't seen this in years maybe. But if we look at abamectin, there's a commercially available product called a racemate that can be injected into trees. It's very similar structure. So if this chemical has activity on these nematodes in animals, maybe these commercially available products in the avermectin class of chemistries could have some activity on the nematodes that cause beech leaf disease. And other ones in the class of avermectin include like Triage, G4, or their R10 product is imamectin benzoate, which is in the same class of chemistry. And going further than that, pine wilt disease is another disease caused by a nematode. It affects exotic pine trees in the U.S., where this nematode, Bursa aphelinchus xylophilus, is native. But in Asia, it's been introduced, the nematode has been introduced into Asia, and it's wiping out their forests. Millions and millions of trees have died from this nematode over there. And so some Asian researchers looked at what can we do to manage this. And so they did research on imamectin benzoate, and they injected trees and then monitored them after they inoculated with the nematode over years to see how many years of protection you get. So they had different locations, and they had different pine species that were native there, and they injected them with 10 grams per cubic meter of volume. So what the labels are labeled at for pinewood nematode in the U.S. for injections on pines is about 10 mils per inch, to put that in perspective. So what you can see is the survival ratio. That's the most important thing all the way on the right of this table. And when they injected them, they had anywhere from 100 to 90% survival rating after 3 years. So again, we can protect these trees from this nematode that causes pine wilt disease with imamectin. So maybe we should be able to have some efficacy on beech leaf disease. So started to look at this, and in the laboratory, we received samples and we extracted nematodes from them in the lab. And then I treated them with Triage, which is labeled as imamectin benzoate. It's a 4% product, G4 is. And so it equates to about 43,000 parts per million of imamectin benzoate active ingredient. And I diluted that up to 43 parts per million, and then looked at percent survival or percent mortality of nematodes, over the course of a day. So they were extracted, they were treated with these solutions or treated with water as a control. And you can see that the mortality of all treatments happened within an hour. So they were not alive, they had died at these concentrations, whereas the control, the majority of them survived. So we know that imamectin benzoate seems to have some activity against this nematode. It's just a matter of figuring out how much do we put in the tree and what application method do we put in the tree to make it get into enough concentration in the buds where the infections are happening. So we had a trial, which we worked with ArborJet as well as Rainbow Tree to inject trees at the Holden Arboretum, a select group of trees grown in an orchard with triage G4 and triage R10 as well as aracinate, and compared it to a control over time. And we did these injections. One set was done in fall of 2018, and the other was done in spring of 2019. Dr. Lynn Carta agreed to receive our samples and then quantify the nematodes. So she got the symptomatic leaves, and then she cut hole punches out of those leaves and then counted nematodes from the treated versus the untreated. And so what we found as far as the symptoms were concerned, when we looked at the trees in 2019, we found no visual difference in our experimental trees. Visually, they all had very similar level of disease and had a similar disease severity rating, which is showing here. But what we did find was, on average, the number of nematodes that were found in the leaves when treated with imamectin benzoate were much lower than the ones treated with aracinate or the control. And I should point out that the aracinate treatment, there was some phytotoxicity that occurred, and potentially maybe some did not take up product well for other reasons. And there were outliers. You can see the standard error is quite large, but the imamectin benzoate seemed to have on average less nematodes. So we were hopeful of this with the laboratory experiment as well as this injection trial. Well, fast forward to 2020, I wasn't able to go because of COVID-19, but our collaborators at the Holden Arboretum rated our trees again. And this is separating out whether it was a fall injection or a spring injection. And if we look at the disease severity, 1 being healthy, 5 being more than 75% of the canopy has symptoms, we can see that while the trees did not seem to get worse with the fall injection of imamectin benzoate, whereas numerically the aracinate and the control did, there was no improvement necessarily. So even though we had lower numbers of nematodes with the imamectin benzoate trial, we didn't see an improvement. So whether this happens over time or we need to figure out a new method, we're still learning about this. In the spring, a very similar story. The triage G4 seemed to hold its own and not statistically different, but the triage R10 seemed to get a little bit worse. But again, all of this to say there really was no difference compared to the control. So it's not something that we know enough of yet. We're not giving up on it. We're trying new methods. So part of that, and I'll get to that in a second, but I'll get to the preventive trials in a second, but part of trying new methods is we injected per the label as if we were injecting an ash. So we put the product into the tree in a concentrate form, and we know that product works well on preventing emerald ash borer, but maybe it's because the emerald ash borer infests the wood, and maybe the product in the concentrate form, it's real syrupy and viscous, stays in the wood really well, but doesn't move out to the buds or the leaves where these nematodes are, because the nematodes are not found in any woody tissues. Similar with the pine wilt nematode. So maybe diluting it, and that's where our next step is. We have a trial going on with our Cleveland office, and Kevin Lewis was involved with that, where we injected trees either in a concentrate form or a dilute form. And this is a site that doesn't have beech leaf disease yet, but we have a collaborator that works for Bartlett at the Morton, based out of the Morton Arboretum, named Dr. Chad Rigsby, who has an HPLC machine which can quantify and detect the amount of product in different tissues. So over time, we're going to be sending him samples of leaves and buds, and he's going to run it through this machine and tell us, yes, imamectin benzoate is here, and this is the quantity of it. So we can get an idea of how much, compared to a concentrate injection versus a dilute injection, how much product is getting into the areas where the infections are occurring. So that's going forward. We did, however, try to do a prevention trial. We injected very small trees that were about, ½ inch or less caliper, and we used these veterinarian syringes and triage G4. We made small wounds in them with a drill and then put the syringe in, and we had pressure put on that point with a rubber band. And it took about 2 days for these trees in the fall to take up this product. And then our collaborators at the Holden Arboretum inoculated them in the springtime. In addition to these micro syringes, we also used the quick jet air. We made a much larger wound and forced the product into the trees. And we didn't have that great of results. So this graph is showing all of the trees. The treatments are all the way on the right, so the controls at the top. We have trees where they inoculated one bud called Nematode 1, or another bud, which is called Nematode 2, and then it's scored as did disease happen or disease not. And you can see that in all treatment categories, beech leaf disease occurred. Occasionally, buds did not open, they died, and that was probably due to the wounding. And there was some phytotoxicity. These trees were quite small. But in only 2 instances did they inoculate a bud and the leaf opened and there was no beech leaf disease symptom. And that was when it was treated with imamectin benzoate. So again, not a perfect study necessarily, a nice pilot study, and not the best results for this. But we're still moving forward on looking at dilute forms and how much this product is getting there. But this is what we know up to this point. We have another site where we're looking at prevention. There's a site we're working with the Cleveland Metro Parks, Dr. Connie Hausman and Dan Volk over there. They have a site where it's right on the edge of an infection center. So there's disease, you know, where this tree is, where it doesn't have symptoms yet. About 100 yards in the other direction, there are trees that have the disease. So we injected them in the fall of 2019. Here's Chad Klink, an arborist for Bartlett, that is doing the injection. And then we're going to monitor these over time. And again, they're going to monitor them. I'm not traveling due to coronavirus, and so hopefully we'll hear something about what they're seeing in these trees. So we don't know enough yet. We don't have the data. And more recently, this was just last week, I got samples from Cleveland area that were infected with the nematode. They were taken and then put in moist paper towel and then into a cooler and overnighted to me with the idea I can do this bioassay where I have different products and different concentrations, let the leaves take up the product, making a fresh wound on the twig, and letting them suck it up under a fluorescent light. And then we're going to harvest nematodes to count how many are alive and swimming out of the infected areas. We did a pre-count, we're going to do a post-count. And so we tried imamectin benzoate at different concentrations, but we also tried 2 products that are actives from Bayer Crop Science. One is called fluopyram, and this active ingredient does not have a label for trees yet, but it is labeled in turfgrass industry. It's called Indemnify, and they have really nice results with nematodes that feed on putting greens at golf courses. So we're hopeful this active might have some efficacy as well. And then there's some data to support that this active spirotetramat, again, it is labeled in the nursery level, but not in landscape trees, a product called Contos. It is a true systemic, moving both up and down in the tree, and it works well in nursery plants as a drench application. So again, not having to poke holes in a tree might be a good thing. So we're looking at that at different concentrations to determine if in this bioassay in a laboratory environment, there's less alive nematodes in the treated versus our water, which was just distilled water, we can start to explore these avenues a little bit further. But we don't have data on that just yet. So I think I've given you a lot of information. Some of the information, you know, especially with our injection trials, isn't, you know, it's not that their trees are getting worse. And I think that's the big thing, is that we don't see the trees getting worse, and we have some anecdotal evidence from some of our operations where there are trees, featured trees on properties that were injected, also treated with a soil-applied phosphite product, like Reliant, that would be used for Phytophthora root rot or Phytophthora canker management, and a prescription fertilization, that trees tend to hold on. They don't get worse. They don't necessarily appear that better, but the ones that are on the edge line of the woods that aren't injected or treated continue to decline, in some cases die. And so we think that there's something going on. We just haven't quite figured it out yet. So we're trying alternative application methods and other products to explore this. But if, at this point, if there are any questions, obviously, feel free to email me. This is my email address. We can ask them here in the chat. And then these are the people that have worked with these projects. So the Holden Arboretum, Dr. Lynn Carter at the USDA, Cleveland Metro Parks, ArborJet and Rainbow, as well as more recently Bayer. So I appreciate you taking the time. Hopefully you learned something about the biology. This is a continually learning process with this disease as we make more and more observations, and hopefully we'll find something that works well from the management side. I see there's a couple questions in the chat. One from Kevin Brewer asking, would it be beneficial to add carbohydrates to the soil to feed the mycorrhizae? And that's a great question that we don't know. And I made that statement about the mycorrhizae partners, the fungi that are associated with beech, and maybe there's this give and take idea, and that with the disease, the plants aren't giving as much, so maybe the mycorrhizae aren't as active. We don't have any data to support that yet either, but it probably is something that's happening. So it's possible, but I think more than that, we probably need to do something to try to mitigate this nematode infestation primarily. But certainly, if you find something that seems to work, we would definitely be interested in looking at it. There's another question from Tom. Maybe we can teach the purple finches to eat different trees' buds than the beech. Tom, if you want to work on that, that would be fantastic. Yes, if that is the case. Yeah, I know that was a joke, but yeah, we'll still learn more about these purple finches' involvement and the spreading of it, but it seems to hold up and make sense. And obviously, we can't mitigate the finches necessarily, but it'll be interesting to see what their studies do with finding stuff about the birds. Okay, Tom also asks, what about other injectable insecticides that are less viscous? And he gives an example, Bidrin. And I'm not familiar with Bidrin. I'm not sure what that is. But certainly maybe there could be some activity. I know that there are some older chemistry nematicides that aren't used as much anymore in organophosphates. And in agriculture, they use some even before that in the organochlorine, I think, molecules that are really not so good for humans or applicators. And I don't know what Bidrin is, but if it is one of those, it maybe could have efficacy. But again, we'll have to be thinking along the lines of applicator safety and non-target. And that's what is so great about imamectin benzoate and looking at it, is that it is derived. I mean, it's synthetically produced, but derived from soil microbe, and so probably have minimal off-target effects. Tom responds to the Moget product, good for a lot of insects. It is a restricted use pesticide. So yeah, I'll have to look into that to explore it. But yeah, so we'll know more as time goes on, and maybe we'll reconvene and have better results or better news to report. But like I said, trees don't seem to be getting worse. They seem to be holding on, and as time goes on, maybe we'll find new application methods that make it get better, and that would be the hope. So I guess there aren't any more questions. If you think about some while you're dreaming about nematodes this evening, feel free to reach out via email.
Oh, that sounds good. All right. Well, yeah, last chance. If you want to send something over in the chat, please do. Otherwise, yeah, you're welcome to email Andrew directly or somebody here at ArborJet, and do info@arborjet.com would be fine as well. And why don't we wrap that up? Thank you so much, Dr. Loyd. It was a pleasure, and it was a really interesting presentation. So we look forward to seeing more as the results come in.
Absolutely. Thank you.